Am I audible to you? Narayan.
పైననా
కింద గాలి తీసుకోపోతుంది కింద గాలి తీసుకుంటారా బయటోడు
మనకై నియర్లే
రోడ్ కూరలు
మనకై నియర్
వంటకింటి ఏం లేదు పండుకొని వెళ్తున్నారు
ఎంతమంది ఒకటేగా
ముగ్గురు ఇద్దరు వస్తారు మొన్న ఇద్దరు వచ్చారు క్రిస్టియన్ ఉన్నట్టుంది
క్రిస్టియన్ అంట తెలియదంట
అడగలే మంచిగా పిల్లలు మంచిగా కొట్టింది
Good evening, everyone. Is I'm audible? Please check and anyone reply, please. Hello. Is I'm audible?
Yes, ma'am.
Yes, ma'am.
Done. So many of them are late joining. That's why delay. Hmm? Okay. So screen is visible. Okay. So I am Mohna T. Kumar. Okay. Today we are going to learn about, or we are going to discuss about. Don't scribble. Please don't scribble on the screen. Okay, we are going to discuss about the drug development, discovery, and what is about clinical research. The things we are going to discuss. Okay, in the beginning, we can start from the history. Okay, here, ancient time. I'm audible, right? Once again,
Yes, madam.
yeah.
Yes, ma'am.
Ancient time, people what they used there, the people used plants as the medicine. In ancient time, the people used plants as the medicine. If they found a plant having medicinal property, then what they used to do is they used to pluck the whole plant. What they used to do, they used to pluck the whole plant and they used to wash it, mash it, and grind, and they used to take the extract and they used as the medicine. How they used to do in the ancient generation, if they found a plant having medicinal property, what they used to do is they used to pluck the whole plant and they used to wash it, mash it, grind it, and they used to take the extract and they used as the medicine. Okay, like that old generation they followed. After that, while plucking the whole plant, what has happened is the plant generation was destroying. Okay, while plucking the whole plant, the plant generation was destroying. So upcoming generation, what they followed is they used to find out which part having medicinal property. What they found, they used to find out which part having medicinal property. Is the leaf having medicinal property, or the stem having the medicinal property, or the flower, or the bud, or the root. They used to find out which part having the medicinal property. If the leaf having the medicinal property, what they used to do, they used to isolate the leaf along and they used to do the same thing. What they used to do, they used to wash it, mash it, grind it, and they used to take as the extract. Like that, they used to continue. Okay, and next generation, this is the second generation followed like this, and the third generation, okay, third generation what they followed is they used to find out which chemical component gives the medicinal property to that part. Okay, third generation what they followed, they used to find out the chemical component which gives that medicinal property due to that part. They used to isolate that chemical compound along. That we used to call it as the lead compound. That chemical component we used to call it as the lead compound. Lead means it is an impurified form of a drug. We can call it as a lead compound. That is the impurified form of the drug. We can call it as a lead compound. Okay, so like that, still we are continuing the same process. Okay, once again I will say it is a history. In the ancient time, if the people found a plant having a medicinal property, what they used to do, they used to listen. Okay, if they found a plant having medicinal property, what they used to do, they used to pluck the whole plant and they used to wash it, mash it, and grind it, and they used to take the extract and they used as the medicine. This is the first generation followed. And second generation, what they followed is they used to find out which part having the medicinal property, and they used to isolate that part along. They used to, if the leaf having the medicinal property or the bud having the medicinal property, what they used to do, they used to pluck that part along, and they used to wash it, mash it, grind it, and they used to take as the extract. That is the second generation followed. And the third generation, what they followed is, they used to find out which chemical component, okay, which chemical component gives the medicinal property to that part. Which chemical component gives the medicinal property to that part. That we used to call it as a lead. They used to isolate that component. They used to isolate the chemical component which gives the medicinal property to that part. That chemical component we used to call it as a lead compound. Lead compound, it's an impurified form of a drug. We can call it as a lead compound. The lead compound, if we purify it, then we will get the drug. Okay. If we purify the lead compound, then we will get the drug. This is the history of medicine. Is I clear to you?
Yes, ma'am.
That.
Yes,
Now, yeah, that's superb. Now, moving to the drug discovery. Here, the first point is, if we are going to develop a drug. Okay, in between, if you're having any doubts, you can unmute your device and you can ask me. Okay, no problem in that. Okay, if you're having any doubts, please unmute your device and you can ask me. Okay, so now we are moving to the topic is drug discovery. Okay, if we are going to develop a drug or we are going to discover a drug, the first main thing which we want to know is about the disease. The first main thing which we want to know about is the disease. Knowing about the disease means we want to know about the disease characteristic, symptoms of the disease, adjoint disease, along with that main disease, everything in and out about that disease we want to study. That we used to call it as choose a disease. We want to choose it and we want to find out the characteristics of the disease, symptoms of the disease, adjoint disease come along with the main disease. Everything we want to study before developing a drug for that disease. Okay, that is the first point. And the second point is choose the drug target. Choose the drug target means in our world, the drugs are available in different sources. The marine drugs are available, microbial drugs are available, and microbial, marine, plants, in different forms drugs are available. So we want to find out, we want to target it, where we want to take that drug, from where we want to take that drug. That we used to call it as choose a drug target. We want to target it, from where we want to take that drug. Nowadays, the drugs are available in different sources, so we want to target it, from where we want to take that drug. That we used to call, that is the second step, choose the drug target. Next one is identify a bioassay. Identify a bioassay means what? Bioassay means life system. Bioassay means life system. Life system we want to test our drug in a life system. Is the drug is safe, effective, such things we want to test our drug in a life system. Life system means living organism. Okay, maybe it is an organism, maybe it is an animal, maybe it is a human being. We want to check our drug in a bioassay. Bioassay means life system. In a life system, we want to test our drug. That we used to call that identify a bioassay. Next one, find a lead compound. Next one is find a lead compound. Lead compound means what? Lead compound, it is an unpurified. Swati, don't scribble. Next one is find a lead compound. Lead compound means, I already told that it is an unpurified form of the drug. We can call it as a lead compound. Unpurified form of a drug, we can call it as a lead compound. That lead compound we want to find out. Next one is that lead compound we want to isolate and purify. We want to isolate and purify the lead compound. Next, what we want to do? We want to determine the structure. What we want to do? We want to determine the structure. These all are the structure of drug discovery. Once again, I will see drug discovery. We are discussing drug discovery having 6 steps. 6, right? Yeah. 6 steps. First one is choose a disease. We want to find out in and out about the disease. That is choose a disease. Next one is choose a drug target. We want to target it. We want to target it from where we want to take that drug. That is choose a drug target. Next one is identify a bios. In a life system, we want to test our drug. We want to test our drug. Is the drug is working or not? Such things we want to find out. And find a lead compound. Lead compound means what? It is an impurified form of drug. We can call it as a lead compound. And next one is isolate and purify the lead compound. Isolate and purify the lead compound. Then determine the structure of lead compound. These all are the steps involved in the drug discovery. Okay, next one. Any doubts? You can ask me.
Ma'am,
Yeah, tell me.
I'm Geetu, ma'am. This second point, as you said, choose a drug target. Couldn't it be receptors, ma'am?
Once again.
Ma'am, the second point that you said, choose a drug target.
Choose a drug target. We want to choose the drug from where we want to take that drug. From different sources. Nowadays, the drugs are available in different sources. In marine drugs are available, microbial drugs are available, in plants drugs are available, right? So we want to target it from where we want to take the drug. That we used to call it as choose a drug target.
Okay, ma'am. But this could also mean that this drug receptors can also be chosen, no, ma'am?
That is will be different point. Okay, this is different point. Okay.
Okay, ma'am.
This is a drug discovery. That is targeting it. And that point we will come on next topic. Okay.
Okay, ma'am. Thank you, ma'am.
This one is different. This is a drug discovery. We are discovering. Okay, we are discovering. We want to discover. That is the drug discovery. Choose a drug target. Fine.
Okay, ma'am. Thank you.
Thank you so much. Okay, so we are moving to next slide. Okay, so drug discovery we have finished. Now we are going to move to drug design. There are two type of designings are there. There are two type of designings are there. One is target interaction and another one is access to the target. There are two type of designing is there. One is target interaction and another one is access to the target. Target interaction is a structure activity relationship. Target interaction is a structure-based relationship. What is meant by structure-based relationship? The structure-based relationship means muted device, all of you. Structure activity relationship, we can call it as a pharmacophore. It is a medical term. It is a pharmacophore. Pharmacophore means it's a structure activity relationship. A structure-based relationship. For example of a structure-based relationship is a lock and a key model. Only the lock opens when the perfect key is inserted. All of you muted, why? Mute all. Okay. Only the lock is open once the perfect key is inserted, right? Like that. If the disease is there, only the perfect drug is inserted, the perfect drug is intaken, then only the disease will be cured. That is the main example of pharmacophore. It is a structure-based relationship. It is the medical term is pharmacophore, structure activity relationship. Next one is pharmacophore summarize. Yeah. The pharmacophore summarize the important binding group which are required for activity and their relative position in space with respect to each other. This is why I call that it is a lock and key model. We can take it as an example. Pharmacophore is a structure activity relationship. The structure activity relationship, the another term is pharmacophore. The example you can remind is a lock and a key model. Is that clear to you? Yes, ma'am.
Yes,
Done.
ma'am.
Next one is optimizing access to the target. I have told that two type of designing is there. One is target interaction. Target interaction is a structure activity relationship. Okay, two types of design. One is structure activity relationship and another one is access to the target. The structure activity relationship we already studied, that is pharmacophore. Okay, now optimizing access to the target, accessing the target. For accessing the target, what we are doing is the prodrug. We can use prodrug. Here the prodrug, what they are doing is most of the drug having the capacity to make the disease cure. Okay, most of the drug having the capacity to make the disease cure, but some drug does not having the capacity to make the cell permeable. So that time what they used to do, the prodrug will improve the permeability of the cell. The prodrug, what they used to do, the prodrug will make the cell permeable. The main use of prodrug is the prodrug will make the cell permeable. The permeability will be increased if we intake the prodrug. Okay. Now we are discussing about access to the target, accessing the target. So accessing the target, the first point is prodrug. The prodrug will improve the permeability of the cell. Next one is drug alliances. Next point is drug alliances. Here the drug alliances, what they are saying is some drugs, okay, some drugs having the capacity, some drugs having the capacity to cure, okay, some drugs having the capacity to stop the activity of other drugs. Some drugs having the capacity to stop the activity of other drugs. That we used to call it as drug alliances. Maybe some people you have seen, they used to intake multiple drugs at a time. They used to intake multiple drugs at a time. Sometimes it happens. What happened is some drugs, after intaking some drugs will stop the activity of other drug. By intaking multiple drug at a time, what will happen is some drug will stop the activity of other drug. That we can call it as drug alliances. Some drug found to affect the activity of pharmacokinetics. What is pharmacokinetics? Pharmacokinetics means absorption, distribution, metabolism, excretion. ADMA process we used to call it as pharmacokinetics. Okay, so some drug having the capacity to stop the activity of pharmacokinetic activities, stop the activities of pharmacokinetic. That we used to call it as drug alliances. Is I'm clear to you? Any doubts you can ask me. If you're having any doubts, you can ask me. Shall I move to next slide?
Yes, ma'am.
Okay, once again I will explain. Okay, so so we have studied about the ancient time, ancient history. What they used to do, they use plants as the medicine. Okay, some people missed the first section, right? So just till now people are getting admit. Okay, admit all. Here, the ancient time, what the people used to do is, ancient time, if they found a plant having medicinal property, what they used to do is, they used to pluck the whole plant and they used to wash it, mash it, grind it, and they used to take the extract. That we used to call, that is the first generation followed. Second generation, what they followed is, while plucking the whole plant, what is stopping is the plant generation is destroying that. To avoid that destroying, the second generation, what they followed is, they used to find out which part having the medicinal property. If they found the plant, the leaf having the medicinal property, they used to pluck the leaf along and they used to wash it, mash it, grind it, and they used as the medicine, like that. And the next third generation, what they followed is, they used to isolate the chemical compound which involved in that part, the chemical component which gives the medicinal property to that part. They used to isolate that chemical compound along. That is the third generation followed. Still that we are following. While that chemical compound we used to call it as a lead compound. Lead compound is the impurified form of the drug. We can call it as a lead compound. Okay, this is the history. Next one is okay. So drug discovery. Next one is drug discovery. Drug discovery having 6 steps. Choose a disease, choose a drug target, identify a bioassay, and find a lead compound, isolate and purify the lead compound, and determine the structure of lead compound. These all are the drug discovery. These all are the drug discovery. Once again, I will say choose a disease, choose a drug target, identify the bioassay, find the lead compound, isolate and purify the lead compound. Okay, isolate and purify the lead compound and determine the structure of lead compound. These all are the drug discovery steps. Okay, one second. Okay, any doubts you can ask me about this topic. Okay. So choose a okay. If the meeting got ended, please log in again. We can continue this topic. Okay. Is I'm audible to you?
Yes,
Yes,
ma'am.
ma'am.
Okay.
Yes,
Yes,
ma'am.
ma'am.
Okay. Until the meeting is ended, you having any doubts, you can ask me. Okay. In this module, we have studied about the drug discovery. The drug discovery having choose a disease steps having choose a disease, choose a drug target, identify a bioassay, lead compound, isolate and purify the lead compound, and structure determination of the lead compound. These all steps are involved in the drug discovery, and two type of designs are there. First design is target interaction, access to the target, optimizing access to the target and optimizing the drug design, optimizing target interaction and optimizing access to the target. In the target interaction, we have studied about the target interaction. We have studied about the pharmacophore. It is a structure-based relationship. We used to call it as target interaction. And optimizing access to the target. 2 points we have studied. First point is prodrug, and another one is drug alliances. The prodrug means it will be what the prodrug will do. The prodrug will make the cell permeable, and drug alliances will stop the activity of other drugs. These are the two terms we have studied in this topic, in this optimizing access to the target. Any doubts you can ask me. In this topic, if you're having any doubts, you can ask me. Once the meeting is ended, please log in again with the same link. Any doubts? Is I'm clear to you?
How
Yeah,
can we differentiate in vitro and in vivo test, ma'am?
once again?
How can we differentiate in vitro and in vivo test, ma'am?
In vitro and in vivo test. You asking about the difference?
Yes, ma'am.
Okay, this meeting will end up now. I will explain the next module. Okay, next session. Okay.
Okay, ma'am.
Otherwise, it will break. That's why.